I'd like to introduce Professor Chris Martine of Bucknell and on the Susquehanna. Yeah, professor of botany and probably a lot of other things, conservation biology, and now known to maybe the world over because he's president-elect of the Botanical Society of America. So they do have a great system with the larger organizations like that where a president-elect will spend what an entire year at that position before becoming president, which makes a lot of sense. So might be something we should consider as well, but we're a lot smaller. So it's going to be exciting times for BSA when Chris takes over. So I became aware of his work when because of his work with a couple of natural heritage botanists in Pennsylvania in 2017, where they were rappelling down a 350-foot cliff on the, how do you say it? Shalami or something like that. It's on the Susquehanna as well. And Chris does some really high production videos called Plants Are Cool Too. I learned about the project from I think reading it in some papers, but the videos that he does, Plants Are Cool Too. You can check it out. The one that's on that bluff starts with the title of something like "Rappelling Down a 100-Foot Cliff to Find Rare Plants." And what it showed was that there's a lot of questions that can be answered when you do that kind of field work. So, and then since I started following MIC, he would take his whole class wading across shallow portions of the Susquehanna to get to an island to do botanical field inventory. So I thought, this guy is really great. We got to get him here. And in the more recent times, he'll tell us about it soon, some of the fascinating work with Solanaceae in Australia. So without any further ado, please welcome Dr. Chris Martine.
Thanks, everybody. I want to apologize about my voice. I lost it on the ride up yesterday, and so I spent the whole day in the herbarium trying to be really quiet, and so I could save it all for now. So I'm ready to use it all up. Can you hear me out there? That's 500 people on Zoom. That's 500 people that all just yelled yes. They say that because my mom's probably listening, and I want to make sure she knows there's that many of you in the room. All right. So let's talk about some solanums from Australia. I know this is the New England Botanical Society, and I've actually been a member of the society for a really long time, since I was a grad student at University of Connecticut back in the beginning in 2001. Today, I'm not going to say very much about New England plants, but I am going to talk a lot about Australian plants. We're going to talk a lot about sort of the value of studying any group of organisms closely, and the things that you can learn, and the lessons that you can learn, and the ways that you can sort of teach and engage students in some of these same lessons, right? And then also a little bit about how we've parlayed some of these interesting outcomes related to plant reproduction into science outreach and science communication kind of outcomes that I want to share with you today. I'm going to use the term we a lot during this. And when I say we, I mean myself and my lab group. I'm on sabbatical this year. So we've got sort of a small but mighty group in the lab right now, and I'm not around a ton, so that's a good thing. We graduated a bunch of great people last year. We've got some folks that are sort of spending time working with me closely, and you'll hear about some of their work today. I've also been really lucky to be able to fund postdocs in my lab since I got to Bucknell 14 years ago. And so we includes these amazing people, but I really want to make sure you know that it includes a lot of undergraduate students. Bucknell is a primarily undergraduate institution. It's like the sweet spot for me. I was just saying this to Ryan earlier today. And if you are somebody that's a grad student or a postdoc and you're thinking about a career in an undergrad institution, hit me up anytime. I'm always happy to talk about this sort of stuff, right? Now, I do actually, as Bob mentioned, care about the flora of Eastern North America, right? So I do a lot of work in Eastern North America, and I'm not going to say much about that today. That includes let's see if I can actually hide this without shutting down the entire operation. Still working? Great. All right. So we do a lot of conservation work with rare plants of the Central Appalachians, conservation genetics, and other things. So I'm not going to say much about that today. I'm really rooted in the Northeastern flora personally. I've been really lucky to be able to work on this publication since I was an undergrad, actually. So there's 7 editions of this that we've now published since that time, and the companion guide, Shrubs and Vines of New Jersey and the Mid-Atlantic states, my wife and I worked on, and she illustrated everything in this guide of about 114 or so species. So, and I was really pleased that our university, she works at Bucknell, also promoted this recently with our 3rd edition of the book that came out. So I really am interested, and I became a botanist in the Northeast, right? And then I've applied those things that I've learned now to other things that I've been lucky to study. What I am going to focus on today are these solanums of Australia. And really, for me, it's this fascinating reproductive biology that these things do, including all sorts of things related to their sex expression and the variation and diversity in that sex expression. And in particular, in this group of things that are called bush tomatoes, even though they're actually more closely related to eggplants than anything else in the genus. So I'm going to talk a lot about reproductive biology in this particular group of plants. I know everybody knows they're parts of a flower already, but I want to sort of put it in the context of this group so you know when I'm using terms what I mean. I'm going to be using the term cosexual a lot. I mean that as a synonym for things that we may have heard as hermaphrodite or hermaphroditic or bisexual or the perfect flower. And this means, you know, we've got all the capacity to do both male and female stuff in that flower. I'm going to use the term male a lot, right? Even though technically we might use the term staminate, right? And this is to sort of represent the sum of all male parts, the andrecium. I just mean that these are the organs that are going to produce sperm cells and make pollen grains, right? And I'm also going to use the term female throughout the talk. Of course, I do mean the female parts, the gynecium or the carpellate organs, or maybe the pistillate organs, right? Just the point being that these are egg producing and seed producing organs, all right? So that's the typical Australian solanum flower, this nice, beautiful purple corolla, right, of the flowers with a nice yellow anther cone. So I'm studying variation in the way these flowers are presented in a particular group of plants that are called the Solanum dioicum plus Solanum echinotum clay. This is about 45 different species that live across the Australian monsoon tropics, sort of this upper third of the continent. We get 3 months or so of monsoon rain, downpours, lots of flooding, and then 8 or 9 months of almost no precipitation at all. I've been on multiple trips to Australia, and I can count the number of days it's rained when I go there in sort of May, June on both hands. But this is not only sort of a seasonal variation, an annual variation that occurs, but there's been a lot of oscillations in terms of climate in Australia over the last 3 or so million years. Lots of sort of very dry, very wet periods, but we've been trending towards an aridification on the continent for quite some time. And my plant group has responded to and continues to respond to that. So we're seeing this big radiation, lots of diversification occurring in Australia as it gets drier and drier. And because of all the interesting ways that these plants have adapted to this, it's a really good model for descriptive study, right? There's all sorts of stuff we don't know about these things. And so I've used that to generate lots of questions that then I can also get students engaged in. They're beautiful plants, right? The flowers are awesome. Lots of typically large fleshy fruits, and then loads and loads of prickles, right? So they're really nice. And so I've been working on this group for a really long time. So what I'm presenting to you today are really like summary statements of more than 2 decades of working on this group. I've been lucky to go to the Australian monsoon tropics for 8 different trips and collect all sorts of plants and make all sorts of observations. So a lot of what I say today in a very short amount of time is a culmination of a lot of that work. I got excited about this plant group as a grad student at UConn with my advisor, Greg Anderson, because of the presence of 3 different sexual systems in this group, right? And so these are the 3 things that these plants do, right? And they do it often in the same place. So when I use the term sympatric here, what I'm saying is that you can often walk into a habitat, you'll find 3 different solanum species, each of them exhibiting one of these 3 sexual systems. And I'm going to go through each of these in terms of what they mean for my particular plant group now, right? So What we think of as the ancestral condition in Australian solanum is this idea, right? Where you've got every single flower on every single plant being co-sexual, right? So they do exactly what you think they could do. Those yellow anthers produce a pollen grain. Is the cursor visible? Yeah. That pollen grain has these openings on it, right? These pores. Through one of those, a pollen tube can emerge and deliver sperm. And now we have male sexual function being completed, right? And that ovary nestled in those anthers, the style and the stigma. Through that, we have the production of flowers and fruits. So, fruits and seeds. So every single flower can do both male and female function. And in my group, there's about a dozen species that do this. We also have 14 or so species that do this thing called andromonecy. And in this condition, we have every single flower cluster, every inflorescence has at its base a co-sexual flower that has both male and female organs. And then above it in the same inflorescence, we get multiple male flowers, anywhere from 10 to 80 up in the upper part of the inflorescence. And typically the only one that actually is co-sexual is the one down at the bottom. These flowers also do what you would expect them to do, right? Each of the floral morphs produces a pollen grain with those pores, and a pollen tube can emerge, deliver sperm cells. So each of the flower types can achieve male function, but only the basal co-sexual flower can actually make seeds and fruit, which makes a lot of sense, right? Now, the third thing that these solanums do is dioecy. And many of us already know what this is, right? This is when you've got separate male plants, separate female plants. We've got really good examples in the northeastern flora, like willows, right? Other plants we know, like cannabis. All the hollies are a great example. So this is one of our, native hollies, Ilex verticillata, the winterberry. On the left, you see male flowers. The obvious organs are the stamens. Those anthers are there producing pollen, and that's going to happen on male plants. And on the right, you have female flowers. And in this particular species, it's also really obvious. You don't see yellow stamens. What you see is a big fat ovary and an almost non-existent style and a big fat stigmatic disc on the top of that, right? This is pretty rare in angiosperms in the flowering plants, right? 5, maybe 6% of all species on Earth do this, but it's also really widespread. So it pops up in multiple lineages all around the planet. Usually it looks like this. You can clearly tell who male plants are and who female plants are. But in solanum, that's not the case. In solanum, we have something called functional dioecy, right? So when you are separate male plants and separate female plants, your males look like this, very clearly male. You don't see a style and a stigma, and they produce that functional pollen grain with the openings, right? But your female flowers, born on female plants, look like this. Morphologically, they're co-sexual. They are producing anthers. Those anthers do produce pollen, but the pollen is inaperture, meaning no openings, right? And that means a pollen tube cannot emerge. So even if you had the right stuff inside the pollen grain, it's still like tossing a pebble on a stigma, right? They just land on stigmas, they get moved around, they get delivered, the pollination occurs, and they can't germinate. Nothing happens, right? So in a functional sense, the flower on the right, still male, but the flower on the left, but the flower on the right is functionally a female flower. And so is the plant that makes those flowers, right? And so this is what we call functional dioecy in solanum. It's the only way that dioecy actually occurs in the entire, across the whole genus, 1,200 species. And when dioecy happens, it's always like this. So another way to say that is we've got male flowers that look male, they produce functional pollen. We've got female flowers that look co-sexual, and they produce non-functional pollen, but can set fruit and seed. When I learned about this as a grad student, I was like, yes, this is what I want to get into. I want to study this for my dissertation. And now I'm, 20 plus years, it's still something I think about every day, right? I'm really excited about this totally cool phenomenon and thinking about why and how, right? So, generally being dioecious seems like a pretty decent idea if you don't ever want to self-fertilize, right? And so Darwin wrote a whole book about this subject, right? This idea that like having separate male plants, separate female plants is a way to reduce the threat of inbreeding when you're an organism that's stuck in the ground, right? So when you can't move yourself around, then just become unisexual, and then you're always going to outcross, right? So one of the benefits of doing this is that you can't self-fertilize. But one of the biggest challenges to this is the same exact thing, right? Now you can never self-fertilize. When you're the one male on a hill, you're going to die on that hill, right? That's it, right? There's no one to reproduce with, right? So it's a risky thing, right? So the hypothesis forever since that Darwin wrote that book, right, is that it must be pretty important to avoid inbreeding if you're going to sort of take the step. Now, there are other hypotheses about how these kind of things would have evolved, right? Things related to resource allocation. Males are pretty cheap. They can live in sort of poorer conditions. Females need to invest a lot more energy over their lifetime to make fruits and seeds so they can sort of only live in the more optimal conditions, right? There's lots of other ideas surrounding this, but this is one. So that's a little bit of sort of like the like, why would you do this, right? But in the case of solanum, why are you keeping these parts, right? That's a lot of energy to not only make stamens, but also fill them with essentially fake pollen, right? Why would you do this, right? Why would you do this if you're sort of, I'm personifying, right? But if you're committed to being unisexual, to being female, why would you still go through the trouble of doing this, right? And all your brains are already working through it, and you know it's related to pollination, right? So in the genus Solanum, 1,200 species worldwide, no species of Solanum make floral nectar. There's no floral nectar production, right? It's all about pollen feeding bees, right? The pollinators that do the work here in the whole genus are bees that are attracted to these flowers, harvesting pollen, eating it themselves, but also importantly, feeding it to their broods. If you don't have anthers in those flowers, they won't visit, right? That's the cue to get them to come to the flower. And if you don't, then bees are smart, right? We think they can learn, right? So we know they can learn. If you don't fill those anthers with some kind of reward, they may not come back, right? So this is a really sort of intricate way of being unisexual, but also maintaining pollinator fidelity, right? In this system, you can't be female unless you also just keep the male parts around to make sure that pollination occurs. Again, fascinating kind of stuff, right? When I learned about this, it kind of blew my mind. This is the only phylogeny I'm going to show, and I'm only showing it so you know that this is work that we're doing in the lab. This is led by Angela McDonald, former postdoc, who's now a professor at St. Cloud State. This is a 353 gene phylogeny, and I just want to point this out to you that the group that I study is in the orange box. The three different sexual systems I just described are shown here. Green is Andromonaceae with the co-sexual flower in the males. Yellow is Dioeciae, the separate individuals, right? And then purple is co-sexuality. I just point this out because I want to show you that we have some really interesting stuff we're writing up right now with this paper. But the hypothesis has always been that Andromonaceae was kind of a transition to the dioecious condition, and then that was the end. What we're seeing is that we have an entire clade of solanums that then reverse back to this co-sexual condition. And it's some really interesting stuff that I'd love to talk to people more about later. But I just want to show you this and let you know this is one of the primary projects that we're doing right now. And also show you this as a way to say that this allows us to ask a bunch of other interesting and cool questions. And that's where a way for me to now transition to some of the interesting student related and other collaborator related projects that we're doing. So the one that I'm not going to say much about is the one on the bottom right, despite the fact that Diamanda is actually the only one of these students who's actually on Zoom right now listening to the talk. Dia is now at NC State working with Rafael Guerrero, and she's looking at the sex expression stuff and really at the whole genome level, right? What happens in the genome to turn male stuff on or off or female stuff on or off? I think in the next few years, there's going to be some really groundbreaking and cool stuff there. I am going to mention these other three things because I have a few recent findings to share that I think are pretty interesting, and they give a sense for the kind of work that is generated by this sort of larger, bigger set of questions that then can be used to engage students in sort of descriptive biology and other things, right? So the first I'll tell you about is let's keep talking about the pollen grains, right? So we've got these sort of fake, in quotes, pollen grains that are being produced. We know they have to be there in order to get bees to visit, but I've always wondered if those are actually equivalent pollen grains, right? We know that morphologically they're kind of different and functionally they're kind of different, but do they offer the same reward to bees? Wouldn't it be interesting if the fake female pollen was also like less nutritious and wasn't really worth it? And then the bees are basically being completely tricked into harvesting that pollen. So Jackie, who's now Dr. Jackie Indem Galbert, just finished her PhD in cancer biology at UTH Zurich when she was a master's student with me, she was really interested in proteins and amino acids, which is what she went on to do as a PhD student. And she asked if I had anything I'd want to do in that area. And I said, yeah, actually, let's compare these pollen grains. She found, this was published in AJB a few years ago, that the male pollen actually does offer a greater reward. This suggests the possibility that the female pollen actually is a bit of trickery, right? So that male pollen actually is worth more, right? So bees visiting and picking up that female pollen, which they have to do to make sure that pollination occurs, are doing it in a way that they're being tricked a little bit, right? So that's an interesting finding, right? So then I was talking to my friend and now collaborator Avery Russell at Missouri State, as a bee behavioral biologist, and he engaged with his own students in questions related to sort of bee choice, right? We now know that male pollen's worth more, but do the bees know? Are they choosing which flowers to visit because they have figured out which things, or are they really being duped, right? And so he ran a whole series of sort of floral choice experiments that are ongoing right now. And the little bit of information I can share with you so far is that they found that not only are the male pollen grains worth more in terms of nutrition, they're produced at almost 6 times as much volume per flower. So male flowers are really pumping out a ton of pollen. Female flowers are producing less pollen, far less pollen, and it's less nutritious, right? So do the bees know? It turns out they don't discriminate at the beginning. They visit each floral morph sort of equally at the beginning. When they approach flowers, they don't choose, right? But after a couple visits, they actually stop going to the female flowers, right? And this is pretty interesting, right? For this system to work, they have to get fooled into going to female flowers at least a few times or you never get fruits, right? But they also smarten up pretty quickly and stop going back to those individual flowers. Is it because they can assess the volume? Is there something about the taste or the smell? We don't know. But so far, this is a really interesting kind of cool finding, right? All right, so that's bee stuff, right? That's one way genes are getting moved around through pollen. We're trying to figure out some things. We have some interesting questions. I've also thought a lot about the other way that plants tend to move genes around, right? Through seed dispersal. We know almost nothing about most of the species in the group that I study, right? Which makes it a fascinating thing in terms of getting into the natural history of these critters. There's one main paper published in 1979 by David Simon, who was kind of like the major solonologist of Australia. And in that paper, he said, well, most of the things in this group are making big fleshy fruits, right? And those are being ingested. So this is endozoochory, right? The endo is inside, zoochory is animal dispersal. So these are things that are being ingested into the body of animals, the seeds being dispersed, right? He said, I think there's also probably a number of species that are practicing epizoochory, right? Sort of on the animal rather than inside of the animal. And in the case of this, we're talking about these things that are trample birds that kind of get caught up in the fur of mammals. And then he mentioned a very rare mechanical type of dispersal I'll describe now called sensor mechanism. Now, first I'll say that these are 2 studies I'm going to present to you very quickly, both done by Abigail Motter, who graduated last year, animal behavior major, joined my lab and said, is there any plant animal interaction stuff you'd want to do? And I said, yes, let's do some seed dispersal. And then she designed 2 really elegant studies. The first was looking at this weird thing called sensor dispersal. What happens here is you have a fleshy berry, just like all these other solanums, but that berry, as it matures, goes sort of past being fleshy and becomes just sort of very hard and dry. It remains enclosed in the calyx. So the sepals of these flowers kind of fuse together and they fully surround the developing fruit, leaving only a little pore at the apex. Eventually, as those fruits dry, there must be timers on these slides. As these fruits dry, a small portion of the fruit breaks off like a little plug. And that plug for a moment kind of sits in that orifice and then eventually falls out. It almost looks like a little acorn top there, right? And then those seeds are now free to be shaken, right? And so these fruits remain in the calyx attached to the plant. And a 6 to 8 foot tall woody plant just kind of sway back and forth throughout the monsoon season. And they're just dropping seeds all over, like those blow up guys at the gas station that are like waving their arms around, right? And they're dropping seeds around all throughout the monsoon season, in the wet season. And this was essentially the hypothesis, but it had not been tested. So Abigail developed a really cool experiment that she set up in our greenhouse, and it's the first time this has been confirmed in the genus. I was going to say that this paper is currently in revision, but 20 minutes before I started, we got the email that her paper has been accepted. So this is her first paper. We're super excited about it. So thanks. Yeah, that's really cool. So that was awesome. Her other chapter, this is her senior honors thesis. Her other chapter was looking at trample bird dispersal. And in this situation, what you have is, yes, you have a fruit that matures to dry and bony, but now instead of staying inside the calyx on the plant, that entire unit, calyx plus fruit, drops off, and that's now the diaspore. That thing hypothetically might then get caught up in animal fur. We don't know if it was true. That hypothesis had not been tested. So we ordered a bunch of animal pelts. She looked in the literature for different ways that you can test the adhesive nature of trample burrs and animal fur. Lo and behold, it was the kangaroo that came out as the stickiest fur, right, for these fruits, which is great because that's actually what lives where this plant lives, like different types of macropods, wallabies, rock wallabies. So that was pretty cool. We're now writing this paper up. I'm sure that she's a lot more incentivized now that her first paper got accepted, right? So, all right, so that's some seed dispersal stuff we've been working on, but it's, again, examples of like cool questions we can pursue, right? The last sort of plant-animal interaction thing that I'll mention, it has to do with ants. So I mentioned about the pollen foragers. Many of you know that solanum is a buzz pollination system. It's not just the fact that bees are collecting pollen, but the way that they do it. They attach essentially their bodies to the anthers. They vibrate at a frequency that is the right frequency for pollen to be released and come onto their body, right? So it's a very tight association between the bees and these flowers. I see a lot of bees on these flowers, but I also see a lot of ants on these plants when I'm in the field. So Caitlin Henry graduated last year and is now in a PhD program at University of Cincinnati in biochemistry, joined the lab and said, I'm really interested in biochemistry. Do you have any things that we could do related to biochemistry? And I said, yeah, I do. These solanums, I mentioned, they don't have floral nectar, but in the field and in our greenhouse collections, as you see here, we'll often have extra floral nectar. This is little globules of sweet, sticky stuff that is exuded on the backs of the corollas, particularly when they're in bud, and also often on the backs of the leaves. And I said, these have been hypothesized as being related to ants that are sort of hanging out on the plants. They're harvesting this extra floral nectar and then they're defending the plants. But nobody's ever tested to see if what's in that extra floral nectar actually is what plants are looking for. And she said, okay, I'll do it. So she does an HPLC test, high performance liquid chromatography, looking at amino acids in this stuff. This is not the full data set, but some preliminary data. And there's two red things here, two red peaks here. One is glycine, and this has been shown to be a glucose flavor enhancer for ants. So when this is present, this is usually evidence that this is an ant-related mutualism. And then over here is a little peak for proline. That's one of the three key amino acids that ants are typically associated with when a reward is something that's sort of meant for ants, again, in quotes. So this is, again, has not been confirmed before in the genus, and now it has been. So we're writing that up right now, right? Now, the sort of my late blooming interest in extra floral nectar led me to a really cool and interesting finding this past summer. So I went back to Australia in May and June, and I had all sorts of field work planned, a number of places I wanted to go to see if we could find some undescribed species. But I decided this time I was going to spend a lot of time in three herbaria on this trip. So I went to the herbarium at Sydney, the New South Wales herbarium. I went to the herbarium at Perth, the Western Australian herbarium. I went to the Northern Territory herbarium in Darwin, and I looked at every single solanum specimen and just looked through them, right? What's here? What's interesting? What's different? What haven't I seen before? And there was a handful of collections from the northern edge of the Tanami Desert, close to the center of Australia, that looked like a species that I knew pretty well, but had some really odd black dots on the leaves. And my first thought was, okay, these are all like mold, right? Some sort of mold thing, right? And then as I looked closer, I realized that the pattern was really similar. On all the things from that part of Australia, those dots were only on the lateral veins, on the undersides of the leaves. So I talked to one of the field, oh my gosh, what just happened there? Sorry. This is very touchy, this new computer. Yeah. I talked to one of the people I was going out in the field with, and he had made this collection, actually, and Kim Brennan. I said, Kim, when you collected the specimen, did you see anything interesting or odd about it? And he said, yeah, actually, those little dots had full of really sweet, very sticky droplets, right? And I said, I think those are extrafloral nectaries. Now, usually when those are present, you can't see them. You only know they're there because the droplets are there. Like across the entire genus Solanum, there's never been visible extrafloral nectaries that are visible to the naked eye. And some of these are as wide as a half millimeter across. And so they're easy to see. And you can see on this image how easy those things are. And so there we were. I was like, okay, I have some sites we can go to. We got to get there. It took us a really long time, 8 days of travel kind of thing with some stops trying to get to this edge of the desert to see if we could go to that spot, relocate the place where these plants were collected, see if we can see the extrafloral nectaries on the plants, and then see if there are ants. And there were. And they were actively harvesting from those half millimeter openings on the veins. And those are extrafloral nectaries. And we have evidence that ants are harvesting from those things, right? They actually ignored me. I've had other times where I've collected herbarium specimens and the ants have been all over me, right? Very aggressive. And in this case, I think they just couldn't be bothered to move away from this resource. So there were enough plants there, and this is partly in collaboration with another former postdoc, Jason Cantley, who you see there. In this habitat, we looked at the specimens. We thought, well, we need to get to this spot, but we could get there and there could be nothing, right? And everywhere you see kind of like silvery blue, that's a plant, and it stretches way off into the horizon, right? So we just happen to go see tons of plants, tons of ants. We had all the characters we need to describe this as a new species. And so we did all the measurements in the field, and I wrote the paper on the plane ride home. I was so excited about this one. It's such a cool and unusual, undeniable new species. So exciting stuff, right? And so I'll use this. Oh, wait, hold on. So this, again, two things interesting here. So one, this is the distribution, right? So in the herbarium collections, most of those specimens were ascribed to Solanum ossicruentum, and that's the distribution now of Solanum ossicruentum. This is now the disjunct distribution of this thing, this new thing we're calling Solanum nectarifolium. And one of the really interesting things here is that this is just one zoom in on I told you I wouldn't show another phylogeny, but this here is a little piece of it, right? I couldn't figure out why our two accessions of Solanum ossicruentum were in two totally different places in the phylogeny. I kept saying, we must have done something wrong. This must be contamination. Maybe I mislabeled the specimen. It turns out it's because they're two different species, right? So the map bears it out. The habitats bear it out. The morphology, these crazy extrafloral nectaries. And a lot of it thanks to like people collecting specimens and putting them in herbarium collections, right? And that's what led us to this. All right, so I'm going to use this as a way to shift a bit to thinking about some sort of outreach and science communication kind of outcomes, because one of the things that I realized was that I've always been interested in telling people about science. I've always been interested in engaging non-scientific audiences, making sure people know that science is important. They should support what we do. And what I have found is that nothing gets more attention from non-scientific people than new species, right? And I'm going to use the term discovery, even though I know they existed before we gave them a scientific name, right? But new species discoveries are things that people get really excited about, because I think most people assume that we know everything out there, and we all know that that is so not true, right? And so we've described a number of new species from Australia in the lab, and I want to give you a couple of examples of how we've viewed these as opportunities to get non-scientists excited about biodiversity and about botany in particular. So new species and sort of SciComm opportunities and some examples of this. So the first one I'll tell you about is this one that we eventually named Solanum watneyi. Now this was a species, that's my family on the top left, wife Rachel, daughter Izzy, son Jackson. We spent about 6 weeks in the outback, my last sabbatical, and we collected a bunch of specimens of this thing that I kind of knew was a new species. And I wasn't sure what we should call it, and I thought about it a lot, workshopped it with my lab group, and it was right around the time that the book The Martian was turning into the film The Martian, starring Matt Damon as a space botanist. And I was like, when are we ever going to get this opportunity again, right? And if you know the story, spoiler alert, if you know the story, he stays alive on Mars by growing potatoes, Solanum, in red sandy soil, just like Australia, right? So I thought we should name this after that character. So the name of the space botanist is Mark Watney, and we decided we're going to name the species Mark Watney after that character. This has become a a species that's really near and dear to my family's heart. That my wife did the illustration for the paper, and then just 2 years ago, my daughter got a tattoo of my wife's illustration. So this species is pretty important to us in a lot of different ways, right? So really, I thought let's just name it, and then I was writing at the time columns for Huffington Post, and I wrote a little thing saying we're going to name this species after Mark Watney, and then we took the plant to the movies on opening night. The whole lab went to the movies. We took the plant. When I got up to the door, the guy said, "You can't take a plant there." And I said, "Is there really a rule?" And he said, "No, there's not." So we took the plant to the movies, and it loved the film. It really loved the film. So that was fun. And I thought, well, that's good. We pulled that little stunt. That's fun. It got some local coverage. And then it just started getting, as the movie got more popular and was actually Oscar nominated, this picked up more and more steam. This is Emma Frawley, one of the students who's a co-author on the paper. And her picture was all over the place. Thousands of news hits all over the world for this species because we kind of connected this to this pop culture phenomenon. So that was pretty cool, and now the species is mentioned in the Wikipedia article about the novel. And I actually interacted a little bit online with Andy Weir, the author of the book. And he told me that Mark Watney would have been so proud, right, to have a species named after him. And so that was a lot of fun, right? So this really got me going, thinking about like next time we name a new species, we got to come up with some other thing to do, right? So we had this other one that we needed to name. And I felt like I really wanted to sort of try to figure out a way to engage younger people, potential scientists, right? So I went to the local middle school, and this is Mr. Catherman, Brad Catherman. He's the teacher that was willing to let me do this. And I had an assembly for 150 seventh graders where I basically showed slides of this plant. Here's all the pictures. Here's where it lives. Here's all the things we know about it. Here's its biology. And then every student wrote an essay where they picked a Latin name and then defended their choice, right? And like basically tried to make a case for it. We ended up choosing a name that a student inspired, right? So we named this thing Solanum ossi cruentum. It's because when the fruits, ossi meaning bone, when the fruits are fully mature, they're hard as a bone. When they're immature and you cut them open, they turn blood red, right? And so this sort of cruentum means bloody. So this sort of blood bone tomato idea. I thought, like, that's fun, right? And we wrote a press release and got it out there. And initially, like, the idea that we had done this with students was kind of the lead. And then eventually it just became that this was a bloody tomato and a super prickly tomato. And like, there was all sorts of stuff. And that got us all sorts of cool press. And we were on a number of different NPR affiliates. Eventually made it on the top 10 new species of 2017, right? All because we did this, right? Otherwise, people wouldn't have really known very much about it. So I was really happy with that outcome, right? As a scientist, I also wanted people to actually care about the science, right? And so what I'll say is that this is just from a couple of days ago. There's been 15,000 views on the open access paper, right? That is not something that happens to my papers, right? Like people are not reading these papers except for specialists in my group, more or less, right? But so for 15,000 people to have actually read the paper or looked at it at least, right? And then there's other metrics like this altmetric score that's fairly high. It just means that's sort of like a buzz index, like how many people are talking about this thing online. There was a time where there were almost 6,600 Google results for that species name, and they were all news stories. So it was really wild, right? It was like a really cool outcome for us. The last one I'll tell you about in sort of a full story is this one. And this is one that's led to a bunch of cool outcomes that were completely unexpected for me. In 2018 and 2019, two different field seasons, we came across this unusual plant. And I mentioned earlier that the species in my group exhibit 3 different sexual systems, right? This, in a single population, was doing all 3. Right? We'd see an individual that was all male, an individual that was only making co-sexual flowers, one that was andromonaceous, right? And this whole combination of things. And I was really confused and not sure, like, what is this, right? What sex system does this thing have? How do we sort of possibly identify what it's doing, right? And so I went back to the herbarium after seeing this in 2019, and I went, like I did today in the herbarium, I went to the indet folder, right? The place where like the things are that people couldn't identify as species. And I came across a collection from the exact same spot we had been 2 years in a row. And again, the guy who wrote the monograph, David Simon, the famous Australian solanologist, had written this long note. Now, I won't read the whole thing, but he says, it's obviously andromonaceous. I've not seen its like before. It's some variant or aberrant. You know, he goes on to say, like, there's no way of knowing what this thing's actually doing. And then he put it in a folder in 1976. And I came along in 2019 and pulled it out and said, what David found to be sort of frustrating, I found to be super exciting, right? The fact that it wasn't any one of those things was really cool to me, right? So we describe this as Solanum plastisexum, plastic sexuality, essentially, right? Does all the things. And we were pretty excited about it. And we wrote a paper and published it. We wrote a press release. And we ended the press release, just like we ended the paper, with this statement. And really, it's just sort of saying, like, this is a great example about how plants don't fit into these like clean boxes, right? There's sort of, it's really difficult to say that plants do any one kind of a thing, right? And so there's all this variation. There's all these gray areas in between things, right? And what's normative is the non-normativity of the whole deal, right? It's this sliding scale. And this is a great example of that, right? And we put that out there, right? And that was enough to get us in the New York Times. So they got really excited about that statement and wrote this big article with color photos and the whole thing. And that, again, got tons of clicks back on the paper, an alt metric that was 5 times higher than the one for oxycruentum, right? It just really took off for us, right? But the thing that came out of this, there's a number of things that came out of this that were unexpected and really fruitful for me personally in terms of professional growth. One was that New York Times article was read by this artist, Erin Johnson. And she said, you know, I'm really intrigued by this plant. And it ties into a bunch of themes that I usually speak about in my artwork, right? So she came out to Bucknell, took photos, took video. She has now had 5 different art exhibitions around the world focused centrally on our species, right? And then all the other sort of things related to that. So that has just been this incredible, cool science and art collaboration that I never imagined was going to happen. I think I was really inspired by this, right? And so then after this, I talked to a friend of mine who's an art professor at Bucknell, and I asked her if she would teach a course with me, co-teach a course with me. And we have this requirement at Bucknell for sophomores. They have to take an integrated perspectives course. It means 2 professors from 2 different disciplines teaching on the same topic, right? And Anna uses a lot of like floral motifs in her artwork, and uses that as a way to examine sort of the ways we communicate about sexuality and romance through the use of flowers, right? And so we decided to teach this course together, where I'm providing all the sort of biology of flowers and sex systems and plants, and she's bringing in the art piece. And that turned into this amazing, really cool collaborative experience, and one of the best things I've ever done as a teacher. For me personally, I really, really enjoyed doing it, right? More recently, this happened. So the amount of cool interactions I've had with people, non-scientists, since plastisexum came around, I sort of almost can't count. But this is just 2 months ago. I Googled the name and this came up on Instagram. So this is a tattoo artist in North Carolina. Somebody brought in a picture of our species and said, I want you to put this on me, right? I want a tattoo of Solanum plastisexum because it's cool and because of what it represents. So that's like this really interesting sort of impact of our science that, again, I could not possibly have expected. But this is sort of the product of sort of putting ourselves out there a little bit and being a little bit willing to sort of think about creative ways of promoting biodiversity science. Okay. We are still working on things like this, right? This is 2 years ago with postdoc Tanisha Williams and friend and collaborator Tanisha Williams. This was a species where we talked about access to nature related to the morphology of the male floral stem, right? Like that was a fun one. And of course, now we've got this new one, Nectarifolium. And I think this one is so cool just based on its biology that I think that's where we're going to focus. On this one, right? But these have been some of the outcomes in terms of science communications that I've been really proud of, right? And I've been proud of my students who've been willing to engage with us and do this work. And I've met some people and collaborated with people that I would not have otherwise. So I have found it to be a really sort of awesome and special kind of outcome. Oh yeah, there they are again. One thing that I'm not going to say very much about is also a relatively new collaboration with an Aboriginal community called the Mardu community in the Western Desert. I have a set of collaborators now at Penn State that are anthropologists, and they've been working with this community for a couple decades. She, through social media, discovered that Rebecca Bird, one of these collaborators, discovered that I was growing this one species that she's been studying the use of and I was only like 50 miles away, and I had a greenhouse full of this plant, and she couldn't believe it, right? So we started to work together. We got an NSF grant to do this work. And we're now looking essentially at the long-term genetic impact of sort of this casual domestication that has happened with this species. It's a seasonal thing that is harvested from the wild, but the processing of it and the movement of the people as they're harvesting has actually set up a bunch of these like almost gardens around these like water well sites, right? Places where the people are camping at night, processing and cooking and drying the fruits. We're actually finding that the populations of plants that have been actively managed in this way for thousands of years are more genetically healthy. They're like more genetically diverse, a lot less inbred because of all of that movement over the landscape than our populations that have not been engaged with in the same way. And so we're working on a couple papers related to that. If you want to learn about this project, as Bob mentioned, I have a video series on YouTube called Plants Are Cool Too. The most recent episode is about that project, so you can check it out. And you can hear all about it and hear from some of the folks that we're working with out in the Western Desert. So please do check it out. This has been a fun project that I've been working on for about 15 years. There's a bunch of mini documentaries that you can check out on different things related to the Eastern flora, but also things in other parts of the world. So check them out. I mostly make them so I can use them in teaching. So I encourage you to do the same thing if you're so inclined. And I'll end with one last piece of Solanum artwork and say thank you, and I'll take any questions if you've got them.
Yeah, sure. You've given us a real example of what I was looking for, some really great science communication as well as really hardcore science. I think in New England, we're a little more constrained in a certain way about the way we express things. And it's fun to have
Jersey.
So I thought that was really fantastic. And I want to second the little Plants Are Cool 2 video that we put is really high production value, about 20 minutes, I would say, in length.
Yeah, they're all like 10 to 15.
Talking a lot about traditional ecological knowledge of the people there in Australia. And there's a lot of good work that's gone on in the States about that recently too. There's a book called Forgotten Fires, which is really a great academic work from an earlier time that talks about traditional ecological knowledge of Native Americans using fire in a very similar way.
Yeah, there's a lot of fire stuff in this interaction. Yeah.
So yeah, let's open it up for questions.
Thanks, Bob. Yep.
Pat?
Go back on finding multisexual, bisexual.
Yeah, the question was, has there been blowback about, the way we've communicated about the sort of plasticity and the sexual expression of that plant? I would say that I saw a lot of interaction online where there was a lot of sort of back and forth kind of stuff, but mostly I've seen a lot of positive stuff related to it. And I think this is one of an opportunity we have as botanists is like, it's the same, like I've had students in the past say, well, in intro bios that are like, well, I'm not really supposed to learn about evolution, right? It's a thing that my family doesn't, right? But when I teach evolution of plants, it seems so less threatening, right? Like you can teach how plants, how natural selection is acted in plants, and it feels like, oh, well, that makes a lot of sense, right? And I think it's a similar kind of deal here. I think that, like, yes, there's a broader sort of idea and maybe a message, right? But I think the fact that it's being introduced in a way that it relates to plants is maybe a little bit helpful, right? So I haven't had too much of that. Yeah, in the back.
I actually wanted to preface this by saying that in my undergrad botany courses, Plants Are Cool 2 was like our Bill 9.
Oh,
so
that's
like,
ridiculous.
Having this plug is like so funny. Anyway, I actually wanted to ask if you had done any sort of genetic research on like perhaps chromosomal differences in co-sexual plants and like their genetic expression.
Yeah, so the question for Zoom is, have we done any chromosomal work or other things about genetic expression of the different sex forms that sound right? Yeah, well, no, we have not. And I think that where Dia that I mentioned earlier, I think that's where her research is sort of headed. From what we can tell, there are no sex chromosomes per se in solanum, but it might turn out to be the case. Yeah, right. It might turn out to be the case, and yeah, and so one of the little secrets I'll share is that the male flowers, even they, if you really dig down into them, have these tiny little vestigial female parts, right? And so even in early floral development, that begins to happen, and something shuts it off, right? And occasionally we have male flowers that actually don't shut it off, right? And so I had a whole set of slides I could have included here about what we call leaky males. So a lot of our males will actually produce fully functional co-sexual flowers, and they can even set fruit. And so it is a little bit of an insurance. It seems like the one male on the hill, that wasn't it turns out not such a great true story, because sometimes males can sort of move through that shutdown and become fully co-sexual. So there's something in there in development that's like turned on or off. And I think it's probably some sort of expression thing, but hopefully we're working on it. Yeah. Yep.
So along with the same line of the question, so have you crossed these 3 different types of sexual reproduction?
Yeah, have we ever crossed across? So Dia, who I mentioned earlier, who's probably still on the call, that was her honors thesis as an undergrad, and it was attempting to do crosses across sexual systems, and you can, right? We were thinking that eventually you'd see this one breakdown and one sort of rise. Well, I think what we found is that in the case of most of these things, when you make a hybrid, there's this really strong maternal effect. So whatever the sexual system is that the pollen recipient had is usually the one that kind of rises. But we did begin to see some weird stuff, right? Some aborted floral types and that sort of thing. So we have tried it. And one of the things about these plants in general is that they all can be hybridized in cultivation, like just by hand pollination. So there aren't a lot of like fully genetically isolated sort of taxa. But in the field, it's probably more about distance and being far apart. But one of the reasons I was so interested is probably where your brain is, right? Is that these things are co-occurring. So you have 3 things all with 3 different sex systems in the same place. And I'm like, how are they maintaining their identities? But something's working there. Yeah. Yep.
Thanks for the talk. I think it's a really amazing demonstration of how natural history inspire evolutionary questions. And I'm wondering, on the natural history side of things, you're clearly working a lot with herbarium data. Are you also working with community science data and like iNaturalist or some other platform?
Yeah. So the question was, are we working with any community science data like iNaturalist for some of the field work? Yes, I have started now. So the past, I'm on sabbatical. I spent 2 full days 3 weeks ago doing nothing but checking solanums on iNaturalist from those regions and doing IDs and verifications and stuff. And that actually really got me thinking about places I want to go. So in addition to the herbarium specimens, which are still, for me, there's a deep history there that's like incomparable, right? In terms of finding cool sites and places to go. There is now this, right? And I have had a few things come up where somebody just popped it in as solanum, and I look at it and go, "That I've never seen that thing before," right? Or it confirms something I saw one place that's now in a second place, right? And now I have another site to go to. So I'm beginning to adopt some of that to sort of find interesting things. Yeah.
That might harken back to the Twitter experience with the Alba.
Yeah.
Mention that briefly.
Sure. One of the videos that Bob mentioned earlier about the Shikellamy Bluffs, and we were on the ropes and looking, is that we came across a heuchera, an alum root on the bluffs that I tweeted a picture of with a bee on it. And I said, "Oh, look at this, it's heuchera." I call it Americana, and pretty neat, right? And Ryan Folk, who's the sort of global expert in the genus, saw it and said, "No, that's actually this thing called heuchera alba. It's only supposed to be in West Virginia and Virginia." But there you have it, right? And so that video that we were making led to then like the discovery of a whole new distribution of this heuchera that wasn't supposed to be in the state. And now we know that Pennsylvania is the center of of abundance for the species. There's more of it in Pennsylvania than any other state. So, and today I went into the Harvard Herbaria and annotated 10 specimens to Heuchera alba. It was not in the collection until this afternoon, and now there's a bunch of it. So, yeah.
So with like these like big showy flowers, we don't really see with like I guess Arcelanums or even the introduced ones. Is that like a convergent trait across the world or a divergent one? I got to visit Brazil twice recently. It was pretty awesome. And both in the northern Amazon, like the southern edge, they had these identical big showy flowers like you showed. I'm just curious.
Yeah.
If that's like a common convergent trait.
Yeah. So the floral sort of the size and color and the corolla, yeah, that and that's actually one of the things that varies the least across these things. And so particularly there's a subgenus of Solanum, Leptostomonum. Those are the spiny Solanums that includes the eggplant. There's about 450 species in that. Those all look pretty similar at the level of the flower, right? A level of the corolla, I should say, right? Because you get sort of, different morphologies in anthers. You get a lot of variation in style and even stigma. But that sort of basic combination of the yellow anthers against typically a violet or bluish, sometimes even white corolla is really standard. I think it's locked in because of the cues to pollinators, right? Bees are seeing everything negative, right? So they're seeing a dark set of anthers against a light corolla. And that cue is what it's all about, right? Because it's so linked to very specific genera of bees, it's just locked in. So when we describe new species, we can't really sort of say, well, this has this flower color and this, right? Like, as those are kind of always very similar. Yeah.
I noticed, as we talked about a minute ago with fire, and I noticed in one of your photos, they were using a controlled burn.
Yeah.
Is controlled burns of particular importance to many of these species or most of them? Or?
Yeah, the question is whether fire management is important to sort of these species in Australia, and yeah, so this whole group that we work on is really disturbance adapted, and so they're edge things. They do a great job, just like some of the ones. The closest relative to these things here is horse nettle, Solanum carol, and NC. Same deal, right? Like on edges, ag fields, like it's just like it gets moved around, does very well. These are the same way, and they have a number of different, depending on the species, different adaptations. Some of them can die back down to the ground, and then they resprout from underground root stalks. Some die completely, but the seeds are triggered by that fire temperature to germinate. There's a couple of them like that like you won't see them in a site for years until there's a fire, and then there's thousands of plants, right? So there's a lot of that fire adaptive nature of these things. Did I answer? What was the question? Did I?
No, it's just the relation of fire to.
Yeah. So I think a lot of it is they're adapted to fire, but they're also poor competitors. So in all of these subarid tropical savannas in Australia, if you let them go, there's spinifex grass and lots of other grasses and then eventually larger woody plants that are going to come in and dominate the system. When you get into a plant community that's like that, really dense, there's almost never Solanum in it, right? And so what we find is that usually the year after a fire you get germination, years 2 or 3 you get full reproduction, a big seed bank, right? And then they get kind of choked out, and then a fire happens and they're back, right? So they're pretty dependent on it. And so this one species, this Wamala species that the Mardu folks are working with is very much like that. It germinates post fire. So as long as there's fires that are being sort of on a, not, I would say low intensity, semi-frequent fires, this thing is on the landscape.
Some of the very first research that showed that smoke itself could help in germination came from Australia. So
yeah,
that's fascinating stuff.
Yeah. You have a follow-up?
Yeah. The note about them resprouting from roots.
Yeah.
I remember you mentioned that these are woody plants. Is that unanimous with your whole focus group?
They are all, I would say, short-lived woody shrubs, except for 2 species that are biennials. Yeah, they resprout the year, they germinate the year after a fire. They're fully vegetative, like mullein or something, or Vasca. And then in the second year they flush out and flower and fruit, and then they die. So there's just a couple that do that, but most of them are these short-lived kind of woody things. So like I mentioned, some of them get tall, like little trees, right? So yeah.
If nectaries bring ants to prevent munching on tissues that are expensive to maintain in water-limited environments, what is the cost of producing all of that nectar?
Yeah, everybody hear that out there? Yeah, that's a great question, and I'm not sure. It would be really interesting to know if they're able to produce more nectar, right, when there's more water present, if they're able to produce more nectaries, if there's more water or nutrients present. But I imagine it's a pretty expensive thing to do. But also, it's really expensive in these environments to grow, to like make leaves, produce leaves, make flowers and fruits, right? So maybe the investment is worth it. And so one of the things I'm really interested in is across the board, right, looking at allocation of male plants versus female plants to all sorts of different things, right? And so one question I have is, right, do males or females make more of this nectar and make more of these nectaries? It might make sense for males to do it because males don't have the energy expenditure of seeds and fruits, so they can't afford to do it. But it also might make sense for females to do it because they are spending so much energy on flowers and fruits, so they need to protect it, right? And so that's a study I'd like to do. Just need a student to say they want to do it.
Let's give a final round of applause.